Hello, my name is Gordon Mitchell Layton, and the date is 11/21/19. Now that we are done with that, we will not need a webcam because we will just need to be looking at the presentation slides instead. So today I'll be presenting on my Unit 2 case connections. The first case today I'll be discussing is Case 3. Case 3 is about a 5-year-old boy who was brought to the pediatrician's office with the symptoms of drowsiness, persistent thirst, and frequent urination, and based off the symptoms, the doctor ordered his blood insulin levels to be measured. After I reviewed the patient's symptom and the insulin results, I came to the conclusion that he had type 1 diabetes. Type 1 diabetes is a disease that affects the beta cells of the pancreas. Beta cells in the pancreas are responsible for the production and release of insulin, so if a patient has type 1 diabetes, his insulin production will be severely lowered. This statement is in accordance with what the lab results showed. So here are his lab results. I just blew them up a little bit. The patient had a baseline insulin level of 0.08, which can be seen right here, and which is greatly below the control, which is around 6, which can be seen right here. After the glucose load, the patient's insulin serum levels varied from the control even more, which can be seen here. It was around 5, and the control is usually around 4, with plus or minus 11. The results from the blood test are good evidence the patient has diabetes because his insulin levels are extremely low, which is a main symptom of diabetes. Another symptom that led me to believe the patient has diabetes is his frequent urination, also known as polyuria. This slide displays an image of the nephron of a kidney. The job of the nephron is to filter the blood and get rid of waste and reabsorb what the body needs. Normally the nephron is pretty good at reabsorbing glucose in the proximal convoluted tubule, which can be seen right here, portion of the nephron, because the body likes to conserve glucose. However, if a person has diabetes and does not produce insulin, which helps activate the GLUT transporters, which help move glucose from the bloodstream into the tissues of the body, then high amounts of glucose will just stay in the bloodstream. When glucose levels are diabetic high, not all of the glucose can be reabsorbed in the nephron, so it builds up in the urine, and the osmotic pressure of glucose draws more water into the urine. So if you look at the slide, usually glucose is reabsorbed right here in the PCT, but if it stays in it, its osmotic pressure will draw more water into this urine that's in here. And when it goes out of the collecting duct into the bladder, it'll fill the bladder more quickly and it will cause frequent urination. The next symptom that led me to the conclusion that the patient has diabetes is frequent thirst, also known as polydipsia. If the patient is urinating excessively from the mechanism discussed, then the hypothalamus is going to signal that he needs more water, therefore causing the patient to be thirsty. The final symptom given that pointed me to diabetes was the patient's drowsiness. If the patient cannot absorb glucose because he's not producing insulin, then the tissues of the body are lacking a main energy source, therefore making the patient drowsy. The case did not state that the patient had the symptoms of glucose in his urine, however, I would expect that he would have this symptom as well. Also, the patient falls in the demographic where type 1 diabetes begins to present itself, which is 5 to 7 years, and the patient is 5. All these symptoms in culmination led me to the diagnosis of diabetes. The next part of the case is the prediction of the patient's fasted glucose levels. I predict that his fasted glucose levels will be elevated to around 6 or 7 millimoles per liter, which is higher than the normal fasted glucose levels of 3.5 to 5 millimoles per liter. I predict this because the patient is not producing insulin, so the glucose is staying in the bloodstream for a long time and not being absorbed by the tissues of the body. The last part of the case is to discuss which enzymes would be activated under the patient's conditions. Since the patient is not able to use much of the glucose from the bloodstream, he will have to produce glucose from another mechanism called gluconeogenesis. Gluconeogenesis can be seen on the slide here along with glycolysis. So on this side is really glycolysis, and then coming back up into the formation of glucose would be gluconeogenesis. One of the enzymes that I expect to be activated is phosphophenylpyruvate carboxykinase, which can be seen right here. The job of this enzyme is to convert oxoalloacetate, which can be seen right here, into phosphoenolpyruvate, which can be seen right here. And that's so that it can bypass the regulatory step of glycolysis, which converts phosphoenolpyruvate right here into pyruvate. The second enzyme that I would expect to be activated is glucose 6-phosphatase. This enzyme helps with the conversion of glucose 6-phosphate into glucose, which can be seen up here. That completes case study number 3. Now we'll be on to our next case study. The second case study that I wanted to talk about was case study number 2. This study examines how glucose uptake occurs in normal cells versus tumor cells and addresses the modifications that take place in tumor cells. So let's first talk about the molecular properties of glucose. Glucose cannot freely move across cell membranes due to its polarity and large size. This means that glucose has to have some sort of transporter to enter the cells of the tissues. There are two main types of glucose transports: facilitative bidirectional transports, which can be seen right here, so like GLUT1, GLUT2, GLUT3, GLUT4, and GLUT5, and the sodium-dependent unidirectional transporters like SGLT1, which can be seen right here. The facilitative bidirectional transporters can be broken down into either insulin-dependent or independent. Insulin-independent transporters allow glucose to freely flow into tissues at any time, and glucose-dependent transporters require insulin to allow glucose to flow into the tissues. The liver uses GLUT2 transporters, which is insulin-independent, so glucose is able to be taken up by hepatic cells at any time. So here's the GLUT2 transporter. The brain uses GLUT1 and GLUT3 transporters, which are also insulin-independent, so glucose can be transported into the tissues from the bloodstream at any time glucose is present. So GLUT1 and GLUT3 can be seen right here. Skeletal muscle and adipose tissue both use GLUT4 transporters, which are insulin dependent, so glucose uptake can only occur in the presence of insulin. Glucose transporters are essential for the uptake of glucose in normal cells, and if glucose transporters were deleted, the cells would lose a major source of energy and likely suffer some damage. However, if a cell is able to overexpress GLUT transporters on its surface, then it could obtain excessive amounts of glucose. This mechanism of overexpression of GLUT transporters is what occurs in tumor cells. Tumor cells that are growing and metastasizing require a lot of energy. The body's best source of energy is glucose, so it makes sense for tumor cells to overexpress GLUT transporters. This overexpression can be seen in the slide where GLUT1 is being overexpressed, right here. In the normal cell, only one GLUT transporter is being produced. However, in the tumor cell, enough mRNA is being transcribed to translate 5 GLUT1 transporters. So only one GLUT1 is being produced here, and there's being 5 produced in the tumor cell. Both the GLUT1 and GLUT3 transporters are insulin independent, which is also advantageous for cancer cells because it allows them to uptake glucose at all times. This extreme uptake of glucose can be seen in the PET scan. Right here in this section around red, here there's an excessive amount of glucose. This high amount of glucose supplies the tumor cells with lots of energy for proliferation and metastasizing. So we're on to our last slide. One would expect the tumor cell to use this glucose for aerobic respiration because that produces the most ATP. However, tumor cells use aerobic glycolysis to supply its cells with energy. Aerobic glycolysis is the breakdown of glucose into pyruvate and the pyruvate into lactic acid even in the presence of oxygen. The reason behind why cancer cells do aerobic glycolysis instead of aerobic respiration involving the TCA cycle and the electron transport chain is still unknown, but I'm going to discuss 2 hypotheses of possibly why. So the first hypothesis is that aerobic glycolysis evolved in cancer cells as an adaptation to harsh environmental conditions so that the carbon skeleton of lactic acid could be used to synthesize nucleic acids, proteins, and lipids while some ATP production still occurs. This slide displays the evidence for the second hypothesis. Pyruvate can either be broken down into lactic acid with the use of just the lactate dehydrogenase enzyme, or pyruvate can be converted into acetyl-CoA using 3 enzymes in the pyruvate dehydrogenase complex, which can be seen right here. Since so many mutations are occurring in cancer, the chances of one of the enzymes in the pyruvate dehydrogenase complex or further downstream in the TCA cycle or electron transport chain being mutated are much higher than a mutation occurring in the single lactate dehydrogenase enzyme. So cancer needs to be able to keep it simple with aerobic glycolysis. So you can see there's only one enzyme involved in this process and there's 3 in this process, so it just makes more sense for cancer to use the more simple process, which can be seen right here. However, no matter which hypothesis is correct, the fact is that cancer cells are at a disadvantage in terms of ATP production per glucose. So it'll need large amounts of glucose to be flown into tumor cells, which occurs by the upregulation of the GLUT1 and GLUT3 insulin-independent glucose transporters, so that the tumor cell can continue to proliferate and rain terror on the body. So that completes case study number 2. Thank you for listening to my presentation, and I hope you all enjoyed it.